my name is Dennis. Today I'll show you how deep UV Raman can be used in virus research. So for the beginning, I will talk a bit about myself. So this is me. I'm a postdoc researcher who works at ICGB and Electra for the last 2.5 years. I didn't include before. I work in 5-6 different laboratories in Croatia, Slovenia, Spain, and Switzerland. Mainly topics were related with viruses and spectroscopy, but some of the topics were real out. Cancer research, fungi, bacteria, developing biosensors, AFM, redundant stuff. But let's keep it simple. Mostly it's related with virus research and spectroscopy. So for you who is not familiar what ICGB is, it's an international organization that operates under United Nations. Here you can see all the thank you. Okay. So ICGB, on the map you can see all the member states of who are in this network, and ICGB has 3 headquarters. The main one is here in Trieste, 20 meters from here. The biggest one is in New Delhi, and the third one is in South Africa. And I'm not sure if they open in China also, I believe. So more specifically, I'm a member of a molecular virology group. We are interested in basic and applied research of highly pathogenous viruses like SARS, Zika, Ubuntu, biosafety level 3 microorganisms. So about synchrotron, who is not familiar, these are particle accelerators. There are around 50 in the world, and 1/3 are located in Europe. And you can see only 4 places in Europe have both this synchrotron, which is a circular particle accelerator, and free electron laser, which we can say it's a linear accelerator, and 2 of them are located in Italy. The one here in Bazovica attracts users from over 50 countries around the globe. The majority is related to material science, and research dedicated to life and medical sciences are around 12%. Both ICGB and synchrotron are part of a bigger consortium, pathogen research platform named PRP-CERIC, which is dedicated to study of emerging pathogens. Myself, I work and collaborate in 3 different beamlines at Terra Fermi, which we exploit the use of terahertz, CC infrared, and use, which is the topic of today's presentation, deep UV Raman spectroscopy. So here I will just stress you some of the aims of ICGB Electra collaboration. So my main tasks are to establish methodologies and workflow to study viruses, highly infectious pathogens, to elucidate their biological and structural characteristics, try to build a reliable prediction and classification tools to assess possible dangers and behavior of such viruses, and to use non-ionizing radiation technologies like terahertz to deactivate the virus. So here I will describe you for this particular project, the project workflow. So at ICGB, I propagate and purify the virus, lentiviruses. I create liposomes, I prepare different kind of samples. I transport it to Electra, and I use beamline. I do spectral acquisition of these samples. So here is the setup, how deep UV Raman equipment looks at used beamline. So deep UV Raman spectroscopy is a spectroscopic technique that used to detect vibrational, rotational, and other states in molecular system and molecules that are really resonant active in deep UV rays are nucleotides, aromatic amino acids, and lipids. For this study, I'm using excitation wavelength 213, which this wavelength specifically at this wavelength, the molecules that are really active are lipids and proteins. I have another studies big of 266, for instance, that nucleotides are really resonant active with this excitation wavelength. So, okay, let's focus. The excitation wavelength that I use in this study is 213. As a model virus, I used vesicular stomatitis virus. It's a biosafety level 2 microorganism. It's an envelope virus and single-stranded RNA virus to establish and optimize all the procedure. So I want to stress that this is actually the first ever deep UV Raman spectra of an eukaryotic virus. Once I optimize the aligning procedure and preparation methods, I collect the spectra of this virus during a period of 6 months from 12 different batches. And in a black line, you can see the spectra average of all of them, of all 12 batches. So the second step was to confirm that actually the signals from the virus originates from the virus itself. I immunoprecipitated the virus with an antibody specific to the VSV-G glycoprotein. Once I successfully immunoprecipitated, the spectra resembles all the spectra of the PBS. And the second confirmation step that I used was to assess the infectivity of that samples after immunoprecipitation with a plaque assay, and which confirms that I managed to pull down more than 90% of the virus with the immunoprecipitation. The second step to confirm that I have that the signal originates from the virus was to create it by genetic engineering. And on the left panel, you can see lentivirus that have all the components of the VSV. And when I subtracted the spectra with the experimental one of the VSV, the diffraction spectra came close to 0. I constructed lentiviruses that was missing one of the components, starting from liposomes, lenti without RNA, then fully completed RNA. So once that we confirmed that the signal originates from the virus, I divided this presentation into 2 parts. One is the first part considers about the biochemical characterization of VSV virus to assign the bands, what we see in the spectra. And the second part is about the application part of this study. So how I assign the bands. So first, I was measuring single components of amino acids, nucleotide, lipids, and then, but the workflow goes like this. So I extract, so VSV virus is composed of 5 proteins, VSV, G, L, N, M, P. I downloaded their amino acid sequence and calculated how much phenylalanine percentage tryptophan and tyrosine they have. I did the same for the RNA part. For the lipid part, I mimic the composition of the cells that I use for propagation of these viruses, which are Vero cells. And first I measure all the spec on the left side. On the left panel, you can see starting from above the spectra of amino acids, nucleotide liposomes. Each of the spectra are multiplied with the total mass per virion, which we can see for protein is 74.5%, for RNA is 1.4%, and for the lipid is 21.1%, and I rescale them under the VSV spectrum. So here we can see that the band at 1.6, let's say the first horn is 1610, the second is 1650. The band at 6, the signal here arises mainly from the lipid part, while 1610 from the amino acid part. And this region around 1200 is a mutual contribution of the lipids and protein, as well as the region 1000. The RNA contribution is really low, 1.5%, but can be found mainly in this region from 1500 to 1600. The next step was now artificially, or let's say computationally, create VSV spectra by using single components. So in the lower panel, I grouped the single components in 3 groups: amino acid, lipid part, and nucleotide part. Once I group them, I again multiply with their percentage, the total mass percentage per virion. When I average them in the upper panel, I almost got a perfect match, perfect overlap. So once I developed this procedure, I actually believe, and that's why, Andrea, I contacted you last week, because I needed more lipids, and thank you. I didn't know we will meet here. Yeah, it's strange destiny, no? So I believe that this procedure can be mirrored to other types of enveloped viruses with known protein and nucleotide sequences and known lipid composition of cell type that they emerge from. Thus, this procedure can present a powerful first-step indication tool for virus characterization and classification of BSL-3 and especially BSL-4 types of viruses. Because these BSL-3 and BSL-4 types of viruses are extremely complicated to measure outside of BSL-3 or 4 facilities, like synchrotron, for instance. And this procedure to have an indication how can the spectra look before we arrange the actual measurements can be very useful. So the second task is about the application part. I will show you how deep UV Raman spectroscopy can be used to identify mechanism of action of UV radiation on the virus, and also how the mechanism of action of antiviral treatment on the virus. So let's begin. Okay, I will simplify this. So the first part was to identify spectral markers that are sensitive to UV photodegradation. So in the lower panel, this is the initial spectra of the virus. We then on the second panel here in the middle, so we expose this virus for a period of 20 hours. Spectra was collected every 5 minutes. You can see here there is a rapid decrease there is a decrease, especially decrease in 1650 band, which I thought I maybe forgot to stress it more, which is related to the viral enveloping, and 1610 that is related to the protein and RNA part of the virus. This was easily identified by eye. The regions of interest that are not so easily identified by eye, we compare PC1 and PC2 loading plots. And on the B graph, so all regions of interest, there are in total 6, 7, 8. We integrated area under the curve and of all of them and monitor the change of area over a period of 20 hours. Especially two peaks, the black one and red one, have a very good exponential decay fit score. And then to see what's actually going, what's happening inside of this band, I actually exposed single components like amino acid and lipid that have peaks at this region. So you can, and I was monitoring the kinetic profile of these two components. The black one is lipid, the blue one is amino acid part. So you can see the kinetic profile of the band 1650. There's a rapid decrease of the lipid band of the lipid component, and also we have a decrease of amino acid part. So this is a mutual contribution of lipid and amino acid decrease, while at 1610 we have a decrease in the amino acid band, but increase in lipid photodegradation band, which probably compensate the speed of this reduction, which we see at the peak in the peak kinetic graph. So this is the type of studies that can be done in with UV radiation and identifying spectral markers. So the second part is the similar procedure was used to identify mechanism of action of antiviral on the virus. For this purpose, I use a drug with a known mechanism of action, which my lab mates determine, that specifically targets the lipidic component of the virus. And I also incubated not only with the virus, but also with the single components. So in this graph, you can see how 1650 band got all instantly perturbed after 1 and 2 hours. So the black and the red and the green one. And when I expose the drug with the single components, you can only find this effect on the lipids, which my lab mates also determine. But for this procedure, you need, like, let's say, very fast. In 2 hours, you can get some indication how this drug works. While in this case, there was a collaboration between multiple laboratories, which could take for weeks. And also, I assessed the infectivity after the drug incubation, and in 1 hour, the reduction was from 75 to 90%. So in this case, I want to stress that deep UV Raman can, in rapid, tiny label-free approach, provide us information of effect of antivirals of virus and single components in physiological conditions, which can then be Also, a possible application is to create a database of antivirals with known and unknown mechanism of action can easily be created and classified into their target groups. For instance, if they target lipid, protein, or nucleotide component. And also, this procedure can be a first step, fast and reliable drug repurposing strategic tool. So just a summary, some easy, simple conclusions that the peak at 1650 provide us information about the viral envelope, 1610 mostly about viral proteins and the mutual contribution of proteins and lipids we can find in 1200 and 1000 wave number region. Main contribution viral spectra comes from lipids and proteins. Because they make 95% total virial mass, and the contribution of RNA is relatively low. But their main spectral features can be found in 1600 wave number region. The ratio between 1440 and 1650 tell us about the degree of unsaturation of the lipids used. And here a very general conclusion that prolonged exposure of 213 nanometer laser wavelength induced structural changes in lipids and proteins, having the strongest effect on lipids and virus envelope. Antiviral drugs binds to the lipidic component of the virion, leading to disintegration of viral particles. Some future perspectives, and that's it. Is emerging a powerful analytic tool that can be used for detection and biochemical characterization of virus sample in a label-free and real-time approach without compromising its integrity prior to analysis. Analysis of virus are done in liquid environment, which is very practical for system manipulation if you need to reduce the pH to see the structural changes, to introduce something to the system like drugs, antibiotics. It can be used to, we saw that it can be used to elucidate mechanism of action of antivirals, and in this case, physical disinfection method, and explain the mechanism of activation. And based on biochemical and structural characteristic of many viruses, now obtained experimentally or computationally, which I'm working on now, a classification and prediction tool can be used to assess possible dangers and behavior of newly emerging viruses or even the reservoir animal source of virus that they emerge from, depending on the specific lipids. For instance, that in insects you can find only these lipids and that. Okay, thanks for your attention.